Hi everyone, my name is Barbara Issa and I'm the Deputy Head of a research lab here at Heidelberg University, Medical Campus Mannheim. And today I'm going to talk to you about the research that I've been doing throughout most of my academic life that I'm really proud of, which is the role that cholesterol play in physiological cellular processes such as cardiac cellular contraction. So cholesterol, if you guys think about it, I bet that most of you would think about a really bad lipid that can either clog arteries or cause cardiac diseases. Am I correct? Even though indeed cholesterol is involved in those pathologies, cholesterol is also important for regulating physiology of a cell. So cholesterol is initially synthesized internally by specific organelles and then transported towards the plasma membrane. There, the plasma membrane, cholesterol can bind to other lipids and other proteins, forming specific microdomains. They're similar to little boats, and they're called lipid rafts or lipid microdomains. Those lipid rafts, they're very important because they're highly dynamic, so they can navigate throughout the entire plasma membrane ocean, regulating where and when certain cellular cascades and cellular signaling processes can happen within a cell. One way that somebody can study the role that cholesterol plays in cellular scenarios is by removing cholesterol artificially from the cell. So, if you remove cholesterol, for example, you're removing the glue that is keeping this structure together. So, initially, you have a very organized structure formed by lipids and proteins that are redistributed across the entire plasma membrane. So, if there were cellular signaling cascades that were initially regulated due to the position of these proteins and lipids, they're completely abrogated and disorganized now that you have a redistribution of those across the plasma membrane. So that can cause many consequences to different cell types. And one cell type that I was particularly interested in was a cardiac cell. So the cardiac cell is a cell that is able to contract and relax and to transmit this contraction and relaxation to its neighbors. And the result of that is that the entire heart will contract in a synchronized manner. There are two main things that the cells need to have in order to control the contraction process. So one thing is calcium. So calcium is a very important ion that is more concentrated on the extracellular milieu. So if for some reason the cell has a lot of calcium inside, that can cause cell death. So it's very necessary and very important that the cells find a way to regulate the balance of the amount of calcium that is going inside of the cell and the amount of calcium that is going outside. So this is one thing that it's important for cardiac cellular contraction. Another thing is that there is something inside of these cardiac cells that contract and expand. So they're similar to springs, right? That can contract and relax, contract and relax. Those structures are called myofibers, and they are made of different proteins within a cell. But most importantly, those myofibers, they have to be intact. They're somewhat dynamic because they can deform, but they have to be intact. And another important detail is that this contraction and relaxation is highly controlled by calcium. So now that we all know this information, I can explain the results that I had when I removed cholesterol from cardiac cells. So I noticed that the cardiac cells, even though they were beating faster than a normal cardiac cell with the whole amount of cholesterol, this contraction was not very synchronized. And the contraction, the force that the cell was exerting to do this contraction was lower in the case where the cell had less cholesterol in their in the plasma membrane. So then I investigated why this was happening. So if you think about the beginning of our conversation, so initially there were on the cardiac cells those domains that were encompassing proteins and lipids. And one of these important proteins that were inside of those domains in the cardiac cells are proteins that are responsible for controlling the amount of calcium that is going inside and outside of the cell. Once I took cholesterol out of the cells, what happened is that these proteins, they became distributed in a different way in the plasma membrane. So then they reorganized. And that reorganization caused an imbalance of the amount of calcium that was inside of the cell. So overall, the cells, they had a lot of intracellular calcium. The huge amount of calcium that was inside was causing damage to the myofibers. So the myofibers, they were breaking a little bit and they were not able to transmit the forces across a certain cell and across multiple cells one can think of, right? So then the output that I saw was that the cardiac cells with less cholesterol, they, even though they were contracting faster, the contraction was not synchronized, and the force that was produced during the contraction was lower. So these were my main results that I had for cardiac cells. I'll be happy to talk to you about the science that I've done, focused on cholesterol, and I'm open to any questions you might have. And I'm looking forward to talking to you guys more. Bye.